Welcome back to the playlist on porphyrin biosynthesis. In this video, we're going to look at the physiology and biochemistry of this particular enzyme in porphyrin synthesis, which is called Uroporphyrinogen Decarboxylase. This is one of the enzymes that's going to act on the porphyrin ring once we cyclize it from hydroxymethylbilane. Okay? And I just wanted to remind you that we're ultimately going for the synthesis of heme, which is called a tetrapyrrole. Let me write that down. So heme ultimately is something called a tetrapyrrole. And also to remind you what a pyrrole is, which is shown right here, okay? This molecule right here, this is pyrrole. Notice that it's aromatic, okay? It's a 5-membered aromatic ring, although it does satisfy Huckel's rule. It has 2 pi electrons right here, 2 pi electrons right there, and then of course this lone pair right here is part of the aromatic ring, and so we can essentially draw this resonance hybrid right here. So this is our resonance hybrid. That's kind of messy. My pen's kind of messing up a little bit, but that's our resonance hybrid. And so all tetrapyrroles are going to be they're going to be conjugations of 4 pyrroles, thus the tetra in the name. Okay. There's also another type of ring system called corins, which come from tetrapyrroles, but that's used in vitamin B12 biosynthesis, and that's not done in humans. Although at the end of the synthesis of B12, the corin ring system is going to differ slightly from the pyrrole ring system. Eventually we'll have a whole playlist on that. But in this video, we're just looking at the mechanism of this enzyme, Uroporphyrinogen Decarboxylase. So this right here, just to give you an idea of where we're going, this is what we're ultimately doing. We're going from uroporphyrinogen 3, and we're going to coproporphyrinogen 3, which is shown here on the right. And you should be aware of 4 differences between uroporphyrinogen 3 and coproporphyrinogen 3. Okay? And those differences are as follows. Notice here we have these acetate groups right here. Not the propionate groups, which have the 3 carbons, but the 2 carbon ones, which are acetate groups. Those are located where I'm circling in purple. These are the acetate groups on the uroporphyrinogen 3. In each case here, through the decarboxylation mechanism, which we'll look at, we're essentially going to replace those with methyl groups, which I'll put in blue. So each one of those acetate groups is going to get decarboxylated and turned ultimately into a methyl group as shown in coproporphyrinogen 3. Okay? And this is the enzyme that does it. Oftentimes what you'll see it written as is Euro D, capital D, and that just stands for uroporphyrinogen decarboxylase. Now, there's multiple uroporphyrinogens as indicated by this 3 here. Okay, this is just the most common one, just like there's multiple coproporphyrinogens. This just happens to be the most common variant of them, and it's the one that's used in heme B synthesis. So just keep in mind the heme we're going to ultimately is heme B. Okay, so heme B comes from uroporphyrinogen 3 and ultimately coproporphyrinogen 3. Okay, although there are others. So just some background information on this enzyme. What I want to show you is that the actual decarboxylation of these guys right here, the actual decarboxylation uncatalyzed is incredibly slow. The rate is actually about 10 to the negative 19th per second. That's extremely slow. It's never going to happen uncatalyzed for the most part. But actually what's interesting about this enzyme is that it's pretty much the only decarboxylase that does the reaction without any coenzymes. Pretty much every decarboxylase that we've seen up to this point use some coenzyme like pyridoxal phosphate or something like that. But this one's able to do this reaction 4 times. Notice that there's 4 acetyl groups that we're going to have to decarboxylate. So one cycle of this enzyme, one complete cycle does it 4 times. But actually the rate enhancement, even though there's no coenzyme, the rate enhancement is 6 times 10 to the 24th per molar. That's an incredibly high rate enhancement. In fact, it's known to be the greatest rate enhancement of all enzymes known to man. Okay, so that's a really important thing. The rate enhancement is the highest rate enhancement of all enzymes in nature. Okay, but what we're going to do is we're going to decarboxylate every one of those acetyl groups. And in fact, what it turns out is there's a distinct order in which the decarboxylations occur. Okay, and what I've done for you is I've labeled each of these rings as A, B, C, D, going clockwise here. Now, one thing I'll show you is the C and D rings are shown with these propionate groups next to each other. So the rings where you see the propionate groups right next to each other, okay, like this, not where it's acetylpropionate, but where it's propionate, where these two groups are next to each other, those are the C and D rings, okay? And it turns out that the way this enzyme decarboxylates is from D to A to B, and then finally to C. So that's the order of the decarboxylations for this enzyme. Let's actually look at the organic mechanism of how that occurs. So what I have for you drawn here, just to orient you with my picture, this is the D ring right here. Okay, this group right here that I'm labeling in yellow, that's the R group right here. Okay, the one I'll do in orange, if I scroll up a little bit, this that I'm highlighting in orange and everything that's attached to it, that's the R prime. Okay, and then of course we have the propionate group down here, and then we have the acetyl group right here, which is going to be decarboxylated. Okay, so without ado, let's go ahead and look at the mechanism. Now keep in mind there's this arginine that's in the active site that's going to act as a Bronsted-Lowry acid-base donor. So this lone pair that's on the nitrogen of the pyrrole is going to come out here and form a double bond right there. That's going to force these pi electrons to come over here, and these pi electrons will come out and abstract the proton from the arginine residue, okay, generating this deprotonated version of the arginine. Okay, so now what we've done is, first of all, we've destroyed the aromaticity of the pyrrole ring. Okay, but now what we've done is we've activated this carboxylate right here. We've activated this carboxylate for decarboxylation. Okay, and you'll see how we do that in just a minute. Okay, what's going to happen is the carboxylate is going to collapse into a molecule of carbon dioxide. So this lone pair is going to come in here and form a double bond, which is essentially going to break this bond that I'm going to do in dark blue. So this bond right here now is going to break, okay, because this carbon that I'm highlighting in, I'll do it in red, this carbon right here cannot have 5 bonds. It already has a bond to this carbon. It has 2 bonds to one of these oxygens and another bond to the other oxygen. So when the other oxygen comes in to form the second pi bond, if there wasn't any bond cleavage, you'd have carbon with 5 bonds, so the way you alleviate that is by breaking that bond that's in blue. And when that bond breaks, it's going to force a double bond to form right there, and then these pi electrons come here, and then these other pi electrons kick back onto the nitrogen of the pyrrole. So now we have this bizarre looking structure. We've destroyed the aromatic ring, but now we're actually going to regenerate the aromatic ring. The arginine that was here now in the deprotonated state is going to re-abstract this blue proton from the ring. That's going to force these electrons in here to reform the pi electron system and ultimately regenerate aromaticity. These electrons right here are going to come out and abstract a proton from this hydronium that's held pretty tightly in the active site, and that's going to generate water. Okay, and so what we end up getting is a reprotonated arginine residue, how you would find it at physiological pH. We end up getting a water out, and then ultimately, notice what we get here is we get this methyl group right here, this methyl group that's characteristic of every former acetyl group on the coproporphyrinogen 3 nucleus. Okay, so these 4 methyl groups, okay, these 4 methyl groups right here, these were acetyl groups that were on uroporphyrinogen 3. Okay, and specifically the mechanism that I just showed you applies for every single one of these, but if you really wanted to see what it looks like, you're going to have to rotate the pyrrole ring a little bit to get it into the right mode so you can view it. But I showed you this for the D ring, but it's going to apply for all of them. You just have to rotate it a little bit. Okay, but the enzyme is going to do this 4 times to uroporphyrinogen 3. It's going to do it 4 times to this guy, and then it's going to give you 4 carbon dioxides and coproporphyrinogen 3. Okay, I'll also mention this because it's kind of important. You have to regenerate the hydronium at the end to put back in the active site so you can have it right here. Okay, so what's ultimately going to happen is the water is going to do a proton exchange with solution in order to regenerate the hydronium. Okay, but in any case, that gives you 4 methyl groups where there were 4 acetyl groups on the uroporphyrinogen 3 ring system. Okay, and also keep in mind this, that when we do this cycle, we still have the aromatic pyrrole ring at the end. And it satisfies Huckel's rule because it has 2 pi electrons here, has 2 pi electrons here, and then of course this lone pair is tied up in the aromatic ring. So just remember, this enzyme, uroporphyrinogen 3 decarboxylase, converts uroporphyrinogen 3 to coproporphyrinogen 3, and from there we're going to oxidize it into something called protoporphyrinogen. So that'll be the next video. I hope this video gave you a little bit of intuitive sense of this enzyme, and I think the most impressive part of this enzyme is the rate enhancement, 6 times 10 to the 24th per molar, and that's without coenzymes. So there's no coenzymes involved in this mechanism, only an arginine and a hydronium. See you in the next video.